Hi, everyone, and thank you for joining me today for episode 820 of the People Behind the Science podcast. I'm your host, Dr. Marie McNeely, and I am delighted to have you here for today's episode featuring Dr. Michel Starz-Gaiano. Listeners, if you want to learn more about our show, check out some of our past episodes, find our recommended book list, and more, visit us at peoplebehindthescience.com. And we have another outstanding conversation on the way from today's guest. So I hope you're ready to meet another one of our outstanding people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am thrilled to be speaking with our guest scientist, Dr. Michel Starz-Gaiano. So welcome to the show today, Michel. How are you?
Thank you. I'm great. I'm so excited to be here.
Well, we are thrilled to have you with us, and we're looking forward to learning more about you and your work. But before we dive into all of the details, let me first just give our listeners a basic sense of where you are now and how you got there. So listeners, Michel is a professor and chair of biological sciences at the University of Maryland, Baltimore County. She received her bachelor's degree in biology from the Massachusetts Institute of Technology, or MIT, and her PhD in developmental genetics from New York University. Afterwards, Michel completed a postdoctoral fellowship at Johns Hopkins School of Medicine, and she has received the Donald Creighton Outstanding Faculty Member Award from the UMBC Graduate Student Association for her excellence in mentorship. And Michel, today we're excited to get to know you as a scientist, of course, but also as a person. So to start off with, can you share what do you like to do when you're not doing science?
So I really like to cook and bake. I have a kitchen that looks a little bit like a lab.
Ooh.
I think I like to play around with things in the kitchen. I have a couple kids and a husband, and I like to spend time with them, of course, and I like to travel, listen to music, those sorts of things.
Wonderful. So what are some of your favorite things to cook or bake?
I made really good chocolate toffee walnut cookies recently.
That sound amazing.
I like things with chocolate, and a New York Times recipe for orzo with spinach, so that was on the healthier side, so that was good.
They balance out, right?
Right, exactly.
I love that. Well, it's great to get a little bit of a picture of your life outside the lab, but I know you are doing remarkable work in your research. So let's talk about this as well. How do you describe what you do to people who maybe aren't in your field or aren't in science at all?
We study the cells of the body. We want to understand how cells go to the right place at the right time. And that's something I personally struggle with. I need my phone to tell me which way to go and when to go and how to get there. Mm-hmm. But cells, of course, only have their genes and their DNA to do that job. And understanding why they do that is important, or how they do that is important, because if it doesn't happen correctly, for example, you could have embryos that don't form correctly and those embryos could die, or you could have infants born with congenital abnormalities. So something kind of like a birth defect that can be repaired, like a heart condition or a cleft palate. So we want to figure out what are the genetic controls that allow those cells to go to the right place so embryos can form correctly. Or if you cut yourself and immune cells have to come and fight an infection and heal that wound, they need to know where to go and when. And we know that these kinds of genetic controls also get turned on again when you have autoimmune diseases, or if you have a cancer that becomes metastatic and moves to the body, the same kinds of genes are turned on. So we want to try to identify what genes control this and think about ways that that could improve human health and our understanding of the world more broadly. So my lab uses fruit flies to study this problem, which sounds a little funny, but it turns out that fruit flies have a lot of the same kinds of genes that people have. So we call that conserved genes. And we can use fruit flies and change their genes relatively easily, or change what genes are turned on and off in certain cell types. And then we can see, how do the cells respond? Do they go to the wrong place? Do they not move at all? And the other great thing about flies is they have small tissues that are transparent. So we can kind of look directly in those tissues and see how the cells are moving and how they're responding to the changes in their genes. So that's really an incredibly powerful system. We can grow lots of flies and test lots of mutants and find new genes that are required for that process.
Well, Michel, this is fascinating. And I look forward to getting more information about the kinds of projects that you're working on in this area as we go through our conversation today. But we always like to talk about motivation. I think when you're working on cool research, perhaps that's inherently motivating a lot of the time. But do you have a favorite quote or a saying or something that really inspires and motivates you?
I don't know exactly that it's the most motivating, but I like Louis Pasteur's statement that chance favors the mind, which is prepared. So people kind of abbreviate that chance favors the prepared mind. And I was rewatching the Ted Lasso series recently. He says, as a man once said, the harder you work, the luckier you get. And I think that that's really true. Like you have to put in a lot of work to get these opportunities or observations that you might not see otherwise. So I find that kind of inspiring.
I love both of those iterations of the quote, the idea there. And I think as a scientist, not only do you need this kind of inspiration, the motivation to do great work, but you are able to work with some really remarkable people over the course of your career who can really help you get to where you are today. So when you think about your own career, Michel, are there people who may have inspired you along the way or these figures that you looked up to who really motivate and inspire you even continuing today?
There's so many and I can't name them all. I think I try to get inspiration from lots of different people, but certainly somebody who has been really inspirational and formative in my career was my PhD mentor, Ruth Lehman. She's an incredible scientist. She also works with fruit flies and she gets so excited to figure things out and seems to really enjoy what she does, which is really fun to be around. It's kind of contagious. She will take constructive criticism, but doesn't suffer foolish criticism. And sometimes that's hard to do. And she's also really has been talented at assembling groups of people and building a lab culture that's really positive. So those were really important things for me, developing as a scientist, but also developing my own lab.
I love that. I think these mentors during your PhD, postdoc, your student years when you're learning how to become a scientist can have a tremendous impact. So let's talk about that adventure for you. Where did your scientific journey begin and how did you start getting interested in science?
I was always kind of curious about science and math. My mom, before she was a stay-at-home mom, was a librarian. So we went to the library a lot and we read a lot. So I think in high school, my favorite classes were like chemistry and I liked math. And I actually went to college thinking I would be a math major. That didn't go so well. And I had to kind of rethink what I wanted to do. And I didn't go into college thinking I wanted to be a biologist because I thought biologists studied animals in the wild and that kind of thing. And I didn't gravitate towards that. But I took a genetics course and I think that that really changed things for me to kind of think about how molecular components could inform cells or animals or things like that. And I was really excited for that. So then I got to join as an undergraduate. I did a project lab and incidentally where I met my husband and my best friend. And we were trying to identify clone a human disease gene. And that was so incredible to me. So this was before the human genome was entirely mapped. And we were trying these strategies to identify a new disease gene for the von Hippel-Lindau disorder. We didn't, but of course that wasn't surprising because it was really hard. But because of that, I got to join a lab as an undergraduate. And at MIT, undergraduate research is really important. And that's also been really valuable to me since I've had my own lab, because I think those experiences are also really formative. So I was in David Hausman's lab where we were trying to map the human genome, like literally it was that long ago. But I was just so excited to be trusted with some of that work and to learn new things. That was really fun. And it kind of allowed me to join a lab after I graduated. At that point, I wasn't sure what I wanted to do. I wasn't sure I wanted to do a PhD and everything. So I worked for a couple years in Jacqueline Lee's lab at the Cancer Center at MIT. And that was really great because she was just starting out the lab too. So I got to see how do people get this off the ground. And again, it was fun to have that kind of responsibility to learn new things about a protein that we didn't know about. So in that lab, we were interested in how a transcription factor controlled when cells divide. And we were looking in particular at different kinds of cancer cells. And when we looked in different cell types, we got kind of different answers. And that makes sense because every cancer is a little bit different. Every person's different. But I found that really frustrating because I wanted to know what it was normally doing, not when it was going wrong. And I also learned I did not want to work with mice, but I loved being in the lab. So I went to graduate school and tried some different things there. But I was really excited to work with fruit flies because we could combine these sort of genetic understanding of things with molecular biology and then look at what was happening in the organism normally. And I thought that was just so powerful to get a better idea of what the proteins were normally doing or the genes were normally doing. And at the time, there was a relatively new tool that had been found, which is green fluorescent protein. So that was a genetically encoded protein that you could see. So one of the first projects I had in the lab was to use that to track certain cells moving through the embryo. And it worked and they just danced around and it was the most incredible thing. We recorded it on a VHS tape and put music to it because it was so awesome to watch. And I got just addicted to seeing these little cells do certain things. It was just amazing.
Well, that sounds remarkable. It sounds like you had some really excellent early career research experiences that maybe helped shape your interests and help you decide where to go next. And I think that decision point as graduate school is coming to a close is a difficult one for a lot of people. How did you decide you wanted to be a postdoc and how did you decide where you wanted to go?
So at that time I had a baby and my husband was looking for a job. And so we had some constraints around all of that. And people will give you the advice to change fields for your postdoc. And I really didn't want to do that because I had enough change going on. So I decided to go to a lab where I could leverage a little bit more of what I had been doing anyway. And my husband was coming to the Baltimore area. So I was looking for postdocs around here. So I went to Denise Montel's lab and she was doing really cutting edge stuff on cell migration as well and flies. So there was enough overlap with what I had done as a graduate student, but enough differences that I could learn new and different things doing that work. So that worked out really well for me.
Well, that's awesome. And then as you were wrapping up your postdoc, the next big decision point is what you want to do as a permanent position. How did you find your way there to UMBC?
Yeah, so I was really lucky that I knew somebody here and she was someone I knew at NYU when I was a graduate student. She was faculty here and she was encouraging me to apply here. And she's like, it's a great institution. Of course, a lot of people hadn't heard of it and I didn't know much about it. And she made the point, this is Rachel Brewster, she made the point that it's relatively new. The school was founded in the late 60s. So she's like, we're still building it to be what we want it to be. And I thought that that was so interesting to be part of that opportunity. And when you come here and talk to the scientists here, they're just an incredible group of people. They're very smart and excited, but not arrogant. And they're very collegial. And so I just really fell in love with the environment to come here.
Well, Michel, it sounds like you've found a great fit there at UMBC. And I think it's always interesting to hear about the twists and turns of scientists' career paths. So I guess would young Michel have expected you to end up where you are today as a professor and chair of biology studying fruit flies?
It's funny now because I look back and in fourth grade, we had to do a project on an animal. And all the girls in the class were choosing koala bears and horses and things like that. And I was like, that's lame. I don't want to be like all those people. So I did my report on house flies. And I think that's so funny now that I ended up working on flies for my career. I did find them kind of gross at the time, but also super cool because they did a lot of amazing things I didn't know before I started that fourth grade report.
Well, I love that. I think that's wonderful. And I'd love to dive into some of the details of your research. So, Michel, is there a particular project that you are working on right now that you are just the most excited about and want to share with us today?
I would love to. So we study a set of cells in the ovary of the fruit fly, which is where eggs are produced. And there's this group of cells in there that have to migrate across the developing egg chamber to get next to the oocyte. And they have to be there for the fly to make an egg that can be fertilized. So this little migration, they go like multiple times their distance over a couple hours. is what we're studying. They don't have gravity. So the cells are small and they have to just grab on and pull and push their way through this tissue to get to the right place at the right time. And they move through this uneven set of other cells. So they're sort of pushing and pulling their way through these other cells in between them. So we're trying to understand, how do they know which way to go? And there's a number of different ways they could do that. They could go in different directions, and they really don't. They're very single-minded. They kind of go as a group. There's a cluster of them. There's 6 or so, plus 2 cells that don't do any work. They just tell the other cells to keep moving them. So this is sort of a mixed group of cells. And we know that there are some chemical cues or chemoattractants in the developing oocyte. That's the part that's going to be the egg that oocyte sends out to tell the border cells, the cells we studied, to come to that location. So we know that if you take that away, they don't go there. If you move it to a new place, Denise Montel's lab showed that you could get them to go to that other place. So we know that these cues are important for their movement, but we don't understand how they're tracking them. So we think of that sort of like Hansel and Gretel following the breadcrumbs. They can kind of pick up breadcrumbs along the way to know which direction to go. So the classic model for how cells move in response to that kind of chemical is called chemotaxis. They're moving to where there's more of the breadcrumbs. So that makes a lot of sense sort of in your head. But if the cells can't see past where they're touching, then it's harder for us to understand, how do they know there's more breadcrumbs up ahead? So we're trying to understand if there's other information that's influencing what they do. So at certain locations, our most recent paper that just came out in iScience shows that in some locations, there's not this increase in breadcrumbs, or there's multiple places the breadcrumbs are. We are trying to figure out, how do the cells know to get past those points and kind of keep going forward. And one thing that we found was that they work together. The cluster of cells has to work together to kind of get through those intersection points where the information is less clear from the chemical signals. We can't see the chemical signals as hard as we tried so far. So we were lucky to be able to work with some mathematicians who can make a model of where the breadcrumbs might be. And we can make predictions from there about what the cells are seeing or touching. And that can help us understand, how are they using that information. So that would be the next step, is to try to see, are they counting breadcrumbs individually, or do they send information to their friend cells in the back to say, there's some this way, come with me. That's the kind of thing we're interested in.
Oh, that's so cool. And I think you mentioned earlier that you had this interest in mathematics as you were starting your undergraduate education. So was it exciting for you to actually be able to incorporate this mathematical modeling into your research?
I've been so fortunate. It was absolute serendipity that I ran into our collaborator at the first day of faculty orientation. And we started talking at lunch. So that's Brad Piercy. And he had a friend who worked on something similar to what I do in fruit flies. And so he was like, oh, yeah, let's talk more. And so a number of things happened, but we've been working together now for 15 years. And he and his group of students have been fantastic about making sure we understand the mathematics at a level that I can understand, because I didn't do much degree there. And we make sure the biology is clear to them. And it's been amazing as a collaboration. Yeah.
Well, it sounds remarkable. And I think this is a really exciting project, but I know there are difficult times in science as well. It's not always fun projects, great collaborations. There are struggles and failures along the way. So, Michel, can you tell us about a time when you really struggled with something, or perhaps you had a major failure? And if so, could you walk us through how you got through that difficult time?
There's lots of little failures all the time in the lab. So you kind of get some practice that way. When I was a technician, I heard a talk from Robert Weinberg. He's a Nobel laureate. And he was saying like 99% of the time things aren't working. And I was like, what? Like, I thought that only happened to me. And maybe it's 99.5 for me. But anyway, so you get used to try again in the lab. But I think when it gets really hard is when you put all of this work into a project and you write a paper or you write a grant and you think this is fantastic, because you really do at that point think that. And if it gets rejected or your grant isn't funded, those are really difficult times. It's really hard not to take it personally. I think a lot of times you have to walk away, or I'll just go listen to some angry music, like Linkin Park or something. Like, it depends on the situation, but maybe just take some time to be upset, honestly, about it and gain some perspective. That was one place where if I went home and my kids were like, let's play a game. And I'm like, yeah, let's play a game because I don't want to think about work and they don't want to think about work with me. So you can kind of do something else or bake a cookie or whatever. And then you can kind of go back. So it turns out in the end that thinking about why people are criticizing your work or why your grant wasn't funded is actually helpful. So to separate out what are the things that you can learn from that process, what are the things you can make better as a result of it not working out, is really what drives science forward in a lot of ways, right? And so I think you can kind of separate what are the criticisms that are not valid and what are the criticisms that really can make your work better. And then just try to figure out and be happy that you have a new, better way to do it, right?
Absolutely. And I think that's a really positive way to go about it. Allow yourself to feel that disappointment, that anger, that frustration, but then go back and find what are the gems that you can pull from these reviews? What are the things you can do to make your work better? So do you have an example of something that you were really excited about, a project that you really wanted to do that wasn't funded?
The project that I was just telling you about, the first time we submitted the grant for it, it wasn't funded. We had to resubmit. I think most of the grants, right, have been that way where the first attempt, there were some issues or people were worried about certain things in the grant. I want it to not be true, but it was true. So we have to come up with better ways. And that's disappointing because you want to start on the work right away. You don't want to keep coming up with different ways to do things. And I think it also really sort of hones your thinking to separate out, like, if the person's criticizing this, is that because I didn't explain it well? What can I do to explain it better? Or maybe that person doesn't know what they're talking about. Sometimes that happens.
Right.
So maybe I can gently explain why they're wrong. But in the end, it's about this communication piece to explain very clearly what you're trying to do and what are the best ways to do it. Why is this system that you're talking about the best way to do it? You have to think really carefully about that. And it doesn't always come right away. So it is helpful to get that feedback and criticism to get new ideas.
Absolutely. And I think you touched on a really important point there, just the value of strong science communication skills. You can't oversell it for these grant applications. It's something that you need to really be able to quickly, concisely, and clearly communicate why your work is important and why the way that you're proposing to do it is the best way.
Exactly. And I think that's where working with students in the lab actually helps me do that better. It was interesting. We were in a partnership program with Gallaudet University for a while. We had a deaf student in the lab. And then the communication piece was really remarkable there, right? We were working through a translator. So thinking about what's the clearest way you can explain this so that this person can get what you're talking about without just writing everything down all the time. It was such a challenge, but I think really did help me think more clearly about how to explain things.
Absolutely. Well, this sounds like the story had a happy ending ultimately, and we love celebrating the victories, the successes in science, because I think it's often a long and difficult road to get there. So do you have another example of a success that you'd like to tell us about, whether it was a big win or even just a small victory that meant a lot to you?
Sure. So we were really excited recently. We had a proposal accepted to work at the Advanced Imaging Center at Genelia Research Campus, which is a Howard Hughes Medical Institute campus. They wanted to help us see the chemoattractant that I was talking about earlier, the breadcrumbs. And we just had an amazing opportunity to work with people there and to use some of the highest end microscopes there to look at our samples. And it was so much work. Like, we worked for a couple of weeks straight all the time, a couple of people from my lab who were wonderful. And I went there and just worked all the time. But we really saw the most detailed images of our cells moving through the tissue. And it's just incredible to watch. So I think that that was really, really fun.
Definitely. And I think these periods of time where you can sort of uninterrupted, just fully immerse yourself in the work, the hours go by quickly.
Yes. I couldn't do it all the time, but
yeah.
You can do it in these short bursts and it's like summer camp. It's so fun.
Oh, absolutely. Well, this sounds like a remarkable experience. And I think these opportunities to go to different places, to use these facilities can really enhance your research and open up potentially new doors for new directions for your research as well. But we love encouraging people, of course, to enjoy their research, but also to take a break and to expand their minds, to broaden what they think about. And one way to do this is by reading. So we recommend that all of our listeners pick up a good book every now and then. So we love sharing recommendations on the show. And Michel, do you have a favorite book, whether it's a science book or a non-science book, that you would like to share with us today as your book recommendation?
I don't have as much time to read as I would like, of course, but I did really like last summer. I read Tomorrow and Tomorrow and Tomorrow by Gabrielle Zevin, I think. And that's sort of a love story about some computer scientists, and it has a really interesting structure. They're video game designers. I thought it was a really interesting book and pretty fun to read. Right now, I'm reading Frederick Bachman's My Friends, and I really do like that book too. I've read a lot of his books, and they're all fiction. They deal with grief and life and that intersection, and in this case, a little bit of art and growing up. I really have enjoyed that book so far. It's kind of funny, but it deals with grief, so that's kind of odd. But anyway, I'm not done yet, so I'm not sure I can officially recommend it yet. But so far, I've really liked it.
Well, we'll put it on the list because the beginning is good, right?
Sure.
Listeners, we'll have to add Tomorrow and Tomorrow and Tomorrow, as well as My Friends, to the page on our website for you to find there if you're looking for a next recommendation for a book to read. And we've talked about some different aspects of your scientific career, Michel, and I think one of the things that people really enjoy about scientific careers are some of the opportunities to travel and to work with scientists all over the world. So do you have an example of a place that your science has taken you that was really memorable?
A couple summers ago, I went to the Collective Movement Conference at the Isaac Newton Institute for Mathematics at Cambridge University. So I was invited to be a speaker at that conference or workshop. And it was mathematicians and even some biophysicists and some animal biologists all coming together to talk about all different types of collective movements. So obviously I'm looking at the cells moving together, but there were people looking at herds of animals moving through the desert and people looking at the biophysics of molecules moving. And so it was really super fun to see people take these different perspectives and try to think about, what can we learn from these other systems that might apply to our border cells. And I remember watching, I think NearGov had a model that he was making of a bunch of ants working together to move a Cheerio.
Oh, cool.
And I thought it was so remarkable, right? Because how do they know where to go and how do they all coordinate to move this one little Cheerio? And I think that does apply to the ways we think about our problems in the cells. So that was really fun. And then it was just fun to be at Cambridge because it's an amazing university, of course. There's so much history there. And I took a tour and I got to see where Isaac Newton's dorm room was.
Ooh.
So if you think about Isaac Newton, like you don't usually think of him as a college kid in a dorm. He apparently built a little cat door for a cat that he liked.
Oh my goodness.
And I just thought that was so fun to see. Here's the building from the late 1600s. That was his dorm.
Well, I think that is amazing. I love going to these cities that have a lot of this history built into them. Were you able to do any other sightseeing while you were there?
Well, we got to travel around a little bit. My husband and one of my kids met me there so we could see London and visit one of the Premier League stadiums and that kind of thing. So it was fun.
Oh, that sounds like an amazing trip. And I think not only do you get to go to cool places, but you also get to work with cool people as a scientist. And I know there are still stereotypes, unfortunately, about how people think about scientists and how scientists are portrayed in the media. So we try to break some of those on our show by just talking about these human moments in science, these funny memories, just the funny, amazing, creative people that scientists are. So do you have an example, Michel, of one of these human moments that may go against some of the stereotypes that people may still have?
So my department where I am at UMBC is a great group of scientists from all different kinds of biological disciplines. And we have a holiday party every year. And every lab basically has to build a holiday ornament only with lab supplies and then write a story about it or like a poem or something like that. It's always super fun to see wreaths made out of pipette tips. And then a lot of times some of the labs will cheat and buy pieces to put on their ornaments. But we've made like a little microscope angel with glass slides, and it has beautiful shiny parts. The light bulb was the little skirt for for the angel, that sort of thing.
Mm-hmm.
So I think it's fun to see people's creativity, and also they're very competitive. So everyone wants to win for the best ornament too.
Is there a prize for the best ornament?
There is. It's a pizza party, as long as you remind the organizers to make it happen.
Well, I think that sounds amazing. And then in terms of the stories or the poems, are those sort of like the backstory of the ornament or how it was created?
Yes, exactly. So often has to do with the lab elves sneaking around and messing things up, or things like that happening to the experiments in the lab, or some kind of holiday miracle when the experiment works or the grant comes through, that kind of thing.
Well, that is wonderful. And I think it does showcase the sense of humor, the creativity in science, which I think are so important when you're solving some of these big questions. So we always ask our listeners to dream big. If we took away the barriers that normally hold you back, whether it's funding, staff, technology, time, feasibility, whatever the case may be, Michel, if you had all of the resources you could dream up, what is the one research question you would most want to answer?
I mean, it is the question I'm trying to answer. If you ask my lab, they would tell you I want to shrink really small and get into the with the cells and walk with them through the place they move so I can see what they're seeing. Not feasible, but it would be interesting. But we really, really want to see the signal that we think the cells are following. So we've tried a lot of different strategies to do that, to label it and to see it. And this is a common problem for secreted molecules in general. People have not been good at seeing them. It just doesn't work very well for a variety of reasons. So we are trying to do things that I feel like are close to possible. I'm hoping to get there still. And I have to say, in the past couple of years, we've made a lot of progress. So I'm hopeful that one day we'll be able to see where this is in space and time. And it'll really help us figure out, are the cells like counting how many molecules they see and telling their neighbors, or if they're just figuring it out in a different way.
I think that's remarkable, kind of getting in the mind of the cell, right?
Yeah, absolutely.
Well, this sounds like a fantastic dream project, and I'm delighted to hear that you are able to work on the questions that you are just most excited about, which is, I think, the dream for a lot of aspiring scientists out there. So if you had to give advice for our listeners, is there one piece of advice that somebody gave you at some point in your career, Michel, that you could pay forward to them today?
I don't think there's one piece of advice, and I'm not good at taking advice. I think I tend to ask for advice and then do something else and then regret that. But I feel like I'm always learning from our cells that we study, and they work together. That's super important. So working with other people is really helpful. Not trying to do things all by yourself. That's hard too sometimes. To slow down sometimes, the cells will do that too. And kind of regroup and figure out what's working and what's not working. And I think there's not always the right time to do something, but there is a time that it has to happen. That is an important thing too. So it may not be perfect. It may not look the way you want, but kind of just going for certain things when the opportunity arises is really important too. But certainly what you said at the beginning, where I'm lucky that I can study a problem I'm really interested in, and that at the end of the day is really important for people to understand, because it's hard, right? And so you need to be doing something that you really think is worth doing to get through the challenges.
Absolutely. And I think we mentioned earlier in our introduction as well that you've won an award for mentorship. And I think being able to help others along their scientific career journey is a really important aspect of life as a professor. So can you share what mentorship means to you and why it's so important to sort of shepherd the next generation?
I think it's like any kind of relationship. You get a lot out of that relationship too. I love working with people in the lab and getting their ideas, get their excitement and enthusiasm, because it also does kind of influence what I do. And so it's fun to work with people who come together with different ideas, and we can collaborate more. I can teach people how to do the kinds of experiments that I know how to do. But in terms of thinking about the problems, it's great to have different insights and points of view and ideas. So I think of it that way, that it's a two-way street.
Definitely. I think you can learn so much from the students who come through your lab, and they can teach you things because they think about things a different way than you maybe do.
Exactly. So that's part of why working in different kinds of groups is so valuable, because I have my perspectives and I do try to get ideas from other scientists and the literature and from art and books and all those things. But it's really fun to just kick back with people and talk about ideas with them. And that's a really great thing that can happen in the lab, is everybody's kind of unified in trying to understand something. And that's fun.
I love it. Well, it sounds like you built a wonderful lab environment for all of your trainees, Michel, and we appreciate you sharing insights with us today. Is there any last piece of advice or perhaps a last note of inspiration that you would like to share with all of our listeners as we conclude our conversation today?
I would say to keep an open mind about what you want to do and be curious. I think I did not expect to be a fly biologist even when I started college. I would have thought you were crazy if you were like, yeah, you would have a career in fly biology. And I wasn't like a digging around in the bug kind of person. But if you keep an open mind and kind of see the strengths and different ways of looking at different problems, that is really valuable. And taking in that information from all different kinds of people can really help you advance your work in new ways, which is what's so exciting.
Well, Michel, such a wonderful note to end on. We appreciate you sharing your insights with our listeners today. If they would like to learn more about you and the work that you're doing, what is the best way for them to do so?
Sure. So I'm at the University of Maryland, Baltimore County, but if you search that under UMBC, that's an easy way to find us. The department is biology.umbc.edu, and my lab, you can find as Starz Lab. So STARZ lab, one word. And I think that's .umbc.edu.
Well, listeners, definitely take some time, check out Michel's lab website and learn more about the great work that they're doing there. And Michel, we appreciate you sharing part of your story with all of us today.
Thank you so much for having me, Marie. It's been a pleasure to talk to you.
Well, it's been wonderful to have you on the show, listeners. Thank you so much for joining us as well. And we hope to connect again in the future on another episode of People Behind the Science.